Welcome to this tour of the PRIME web portal. PRIME is the Program for Research on Immune Modeling and Experimentation, which is supported by NIAID, and which has developed experimental and computational resources, user-friendly models, and educational resources for computational immunology. The web portal provides a single venue from which to access all of the resources that have been developed by PRIME. The experimental focus of PRIME is on understanding the infection of virus in human dendritic cells and the immune response and developing predicted models of this experimental system. However, the resources that have been developed should be of use for experimental immunologists in a wide variety of research areas and for researchers outside of immunology as well. A key aspect of the Modeling Immunity for Biodefense program has been to develop mathematical models that can be presented in form in which they can be used by immunologists who do not have mathematical backgrounds in order to assist their research. The PRIME portal provides access to many of the mathematical models developed by PRIME in this easy-to-use format. The tour will start by an exploration of PRIME-developed models. The PRIME portal currently hosts 10 models that are directed at understanding various aspects of dendritic cell response to virus, intracellularly and autocrine mechanisms and paracrine mechanisms underlying these responses. A variety of modeling techniques have been utilized, including ordinary differential equations, stochastic modeling, agent-based modeling, and integral differential modeling. Most of the models have been published as part of integrated experimental and computational studies to provide insight into the immune responses that are difficult or impossible to obtain with either approach alone. I'll briefly illustrate the access to one of the models, the immune response modeling of interferon beta pretreated influenza virus-infected human dendritic cells. This model and its relationship to actual experimental measurement has recently been published in the Biophysical Journal in February 2010. The schematic of the model can be downloaded from the web portal and is shown here. The model is based on pre-exposure of naive dendritic cells to type 1 interferon and subsequent infection of the cells by virus. The antiviral response and the interferon production that is elicited are predicted by the model. As can be seen in this figure from the publication, the experimental measurements over a 10-hour time course and the predictions of the levels of those molecular components shown in solid line and broken line respectively show a very good correspondence. The model can be simulated by the experimenter via the web portal using user-friendly buttons and menu controls. Simulation of the model of the baseline conditions generates time course predictions of the levels of 8 cellular components, including phosphostat, SOX, IRF7, interferon beta, interferon alpha. If the experimenter wants to predict the effects of experimental manipulations, such as changing the initial concentration of interferon exposure or a knockout or knockdown of a molecular component in the network, this can be easily done via this interface. I'll illustrate, for example, the modeling with changing the initial concentration of interferon. Merely set the initial concentration to which the cells are exposed to a different level. And let's follow, for example, SOX and IRF7, and we will simulate under these new conditions. And we see that the initial and final levels are different, and the predicted time course of the cellular components under these conditions are altered. In this way, all of the components in the model can be controlled easily via this interface in order to test what the effect on the responses would be, and presumably subsequently to confirm those results experimentally. Other models are also available in user-friendly formats. For example, an agent-based model of the single-cell responses of RIG-I and interferon during viral infection are presented with a video demonstration of the model and a downloadable executable. Thus, the PRIME interface offers a variety of models that can be easily accessed and simulated. Next, I would like to demonstrate the tools and resources that are available via the PRIME web portal.